Hi everyone, welcome back to Path Demystified. So, from today, I have decided to start a series of short tutorials which will take us into in-depth knowledge of a very interesting, a little bit complex topic called neoplasia. We shall go into the concepts of neoplasia, hallmarks of cancers, mechanisms revolving around it, and many more things. And I'll try to make it as simplified for you as possible. So let's start with the very basics. Today we are only going to understand certain definitions, nomenclature, what are benign tumors, what are malignant tumors, and get familiarized with certain terms. Okay, before going into the theoretical definition of neoplasia, let us understand a few terms. The first is tumor. Now, tumor literally means any swelling. It was first described by Celsus and he gave this term in context with inflammation. Celsus gave 5 cardinal signs of inflammation, if you remember, and it was in this context that he used the term tumor to describe any swelling. Second is neoplasia. Neoplasia means new growth. Third is oncos. Now this word oncos was first described by Claudius Galen. He used this term to describe tumors which were different. He distinguished tumors into 3 categories. First, according to the nature. Second, exceeding nature. That means pus producing masses. And third, any swelling which was contrary to its nature. And this is what we now call as neoplasms or new growth. And oncology. Study of these tumors is called oncology. So now we know that neoplasia in simple terms means new growth. But what is the definition? It is very difficult to describe neoplasia, and the only definition that comes little bit close in defining neoplasia was given by an Australian scientist in the pre-molecular era. Rupert Allen Willis describes neoplasia as an abnormal mass of tissue, the growth of which exceeds and is uncoordinated with that of normal tissues. And this growth will persist in the same excessive manner even after the cessation of the stimuli which evoked that change. So that was the definition given in the pre-molecular era. But now, with the advent of certain molecular techniques and our understanding of molecular basis of cancer, in today's era, in the modern era, neoplasia is simply defined as a disorder of cell growth that is triggered by a series of acquired mutations of a single cell and its clonal progeny. Now, what are the characteristics of these neoplastic cells? These neoplastic cells have a survival and a growth advantage that in turn results in excessive proliferation, and this excessive proliferation will be independent of the physiological growth signals and controls. Having understood the definition of neoplasia, now let us move further to understand that all tumors will have 2 basic components. Okay, the first one will be the neoplastic cells and the second will be the reactive stroma. The neoplastic cells constitutes the tumor parenchyma, whereas the reactive stroma is composed of connective tissue, blood vessels, and cells of innate and adaptive immunity. What we need to understand is that the classification and the biological behavior of the tumor is based on the type of neoplastic cells, whereas the growth and spread of the tumor is dependent on the stroma. Now, over here you need to understand that the ratio of neoplastic cells is to stroma will vary in different tumors. For example, in some tumors the connective tissue component will be very scant and these tumors will be very soft and fleshy, whereas in others there will be abundant connective tissue, abundant collagenous stroma, which will impart a desmoplastic reaction, and these tumors will be very stony hard and firm. So, all tumors can be broadly classified as benign and malignant. Now, these benign tumors appear relatively innocent, whether it be a macroscopic appearance or a microscopic appearance. These tumors are non-lethal. They are localized at their site of origin. They do not spread or invade the adjacent structure, nor do they metastasize to distinct sites, and these tumors are generally very easily removed via surgery. Whereas malignant tumors, these are aggressive tumors. They invade and destroy the adjacent structures, and they spread to distinct sites, that is, they metastasize very easily, and collectively all the malignant tumors are what we call as cancers. Now the third term is the mixed tumors. These tumors basically arise from a single neoplastic clone, and this single neoplastic clone is capable of producing both epithelial as well as mesenchymal cells. So now, understanding the nomenclature of these tumors, whether it be a benign tumor or malignant tumor or mixed tumors, let us begin with benign tumors. Benign tumors follow the histogenetic basis of classification. To designate a benign tumor, all you have to do is to attach the suffix "oma" to the name of the cell from which the tumor arises. And mesenchymal tumors or mesenchymal cells are the ones who follow this rule very often. For example, if the tumor is arising from fibroblast-like cells, you call it a fibroma. If the tumor is arising from blood vessels, you call it a hemangioma. And if the tumor is arising from smooth muscles, you call it a leiomyoma. And if the tumor is arising from cartilage, you call that as chondroma. Now coming on to epithelial tumors. The nomenclature of epithelial tumors is a bit more complex. How? The epithelial tumors are named according to their cell of origin, or they can be named according to their microscopic appearance, or they can be named according to their macroscopic architecture. The most common term used to describe benign epithelial tumors is an adenoma, which means that the tumor is arising from the glandular epithelium. These tumors may or may not form glands, but more often, they form glands, and therefore they are known as adenomas. You can also name a benign epithelial tumor based on their cell of origin. For example, if the tumor is arising from the renal tubular cells, you call it as a renal tubular adenoma. And on the basis of their macroscopic architecture, it can be called as papilloma when it forms finger-like or warty-like projections which rise from the epithelial surface, or cyst adenomas when they form large cystic masses such as an ovary, or they can also be called as a polyp when they have a visible projection above the mucosal surface, as you can see here. Now, the nomenclature of these epithelial tumors can be a combination of the macroscopic architecture and the microscopic appearance. For example, you can call this as an adenomatous polyp when it is a polypoidal structure on macroscopy, whereas it is glandular forming on microscopy. So now moving on to the nomenclature of malignant tumors. The nomenclature of malignant tumors is essentially the same as benign, with few additions here and there. For example, when a tumor is arising from solid mesenchymal tumors, you use the term sarcomas. For example, when you have a smooth muscle cell tumor, the benign counterpart was known as leiomyoma, whereas the malignant counterpart is called as leiomyosarcoma. When it is arising from the cartilage, the malignant counterpart is called as chondrosarcoma, whereas the benign you were calling as chondroma. The epithelial tumors, when the tumor is rising from the epithelial origin, we use the term as carcinomas. For example, if the tumor is arising from the squamous epithelium, it is known as squamous cell carcinoma. If it is arising from the glandular epithelium, it is called as adenocarcinoma. You use the term leukemias when the tumor is arising from blood forming cells, especially the white blood cells. And we use the term lymphomas when the tumor arises from lymphoid cells or lymphocytes and their precursors. Now, here it is very important for us to understand the difference between the term cancer and carcinoma. Cancer is a common term which is used for all malignant tumors, whereas a carcinoma is a specific term which is used for malignant tumors of epithelial origin. Now, what are mixed tumors? Mixed tumors, they arise from a single neoplastic cell that differentiate into different types of cells. That is, they have an ability to differentiate into both epithelial and mesenchymal cells. A very classical example for this type of a tumor is a pleomorphic adenoma of the salivary glands. You can see that it has epithelial components which are scattered within a myxoid stroma, that is islands of cartilage or bone. Now, both these components in this tumor are produced by a single clone, and this single clone was capable of producing both epithelial kind of cells and mesenchymal type of cells, and therefore such tumors are called as mixed tumors. So now let us understand a few terms before we go to the end of this tutorial. The first is teratoma. The term teratos means monster. Okay, so these tumors are the ones which contain both mature and immature tissues which are derived from more than one germ cell layers. These tumors originate from totipotent germ cells. What are totipotent germ cells? These are the cells which can differentiate into any type of cells, and these cells are generally found in ovary, testes or embryonic rest. So the most common example of a teratoma is an ovarian cystic teratoma, and these tumors contain bone, epithelium, muscle, fat, nerves and many other tissues like neuroepithelium, etc. Now the next term that we have is an hamartoma. Hamartomas are benign. These are basically disorganized masses which are composed of cells indigenous to the involved tissue. What do you mean by that? It means that it is composed of cells which are normally present at the site involved. These basically arise because of chromosomal aberrations, and these chromosomal aberrations are acquired through stomatic mutation. And the last term that we have is a choristoma. What is a choristoma? This term is used for an heterotopic cell of rest. That is, heterotopic means misplaced. For example, if you find a small nodule of a well-developed, normally organized pancreatic tissue in the submucosa of stomach or small intestine, you will call that as a choristoma. Okay, so that's it for now. Today we understood the basics of neoplasia. We understood the definition. We went through some terms such as hamartoma, teratoma, choristoma. We understood how do we actually go about naming benign tumors and malignant tumors. For the next part, we are going to concentrate on what are the characteristics of cells which makes you call them benign, a tumor benign or malignant. What are the differences between benign and malignant tumors? What is metastasis, and what are the ways of spread of malignant tumors? I'll see you soon with all these lectures and many more. Don't forget to subscribe and put on that notification bell, and do share and like this video. Bye. This is me signing out. I'll see you next time.